Good evening. My name is Ruth Plummer and I'm co-chairing this workshop. I'm a clinical academic in drug development and discovery in the UK. So welcome to this CDDF webinar, the application of artificial intelligence in leukemia diagnostics, implication for clinical trials and cancer drug development. So if we could move to the next slide, please. I was going to give you a little bit of background. I mean, many of you will be aware CDDF, the Cancer Drug Development Forum, is a non-profit organization that aims to stimulate interaction between those of us, the stakeholders, who are interested in development of better cancer treatments. And it's bringing together people from academia, pharmaceutical industries, the regulatory authorities, policymakers, and importantly, the patient advocacy groups to try and get those better treatments. So if we move to the next slide to give you some of the technical aspects, these are in the chat as well for you. We've got all our participants on mute to help with the audio quality. Please let us know any problems in the chat function. And there's also the Q&A function. And then my co-chair Axel and I will be helping take the questions forward at the end of the presentation. Also, there's a little clue there on the double clicking on the Zoom screen to move you between full and exit the full screen mode. And then on the next slide, it's just my last slide really to highlight to you that this talk is a prelude to what we hope will be a very exciting workshop. And we also are, in an optimistic way, aiming that this is going to be face-to-face next September in Amsterdam, and a workshop looking at drilling down more on digital tools and artificial intelligence in oncology drug development. We've all used a lot more digital tools over this lockdown period, but we're really going to drill down on how these are going to work in clinical trials, transform patient care, we would hope, and how digital diagnosis can be brought in to how we do cancer drug development. So with that, on my next slide, I was actually going to pass over to my co-chair, Axel Glasmacher, to introduce our expert speaker tonight. So Axel.
Thank you very much, Ruth, and good evening, everyone. We're very pleased that you're joining us for this important webinar. And as you've heard from Ruth, Thorsten Haferlach will be our speaker today, and there will be a presentation of a little more than 30 minutes, and then we have a Q&A that is currently planned for 25 minutes, but if you have more questions, all of us have volunteered to stay longer to make sure that all of your questions are answered, and please start asking them at any time. The Q&A function is open to you. If we go to the next slide, please. It's a pleasure to present Thorsten, whom I know since a long time and also consider a friend. He is one of the world's leading experts in the field of leukemia. He is the head of the Munich Leukemia Laboratory, which he established together with 3 colleagues in 2005. And today it is, I don't think I'll be exaggerating here, it's one of the leading leukemia laboratories in the world, not just by the amount of samples tested, but also by the numbers of papers published. I was very interested to hear from him over the time how he and his group moved into the field of applying artificial intelligence to leukemia diagnostics. And I think what he is going to tell us will be highly interesting in this regard. I don't want to spend more time in kind of keeping you waiting for it. And so let's just go into it. And Thorsten, the floor is yours.
Good evening, everyone. And Axel, thank you very much for your introduction. So I'm happy to be here today, a part of the CDDF program. And my subject today I'll cover is the application of artificial intelligence in leukemia diagnostics, implications for clinical trials and cancer drug development. I have to disclose that I am part owner of the Munich Leukemia Laboratory. And I have to tell you that leukemia diagnostics aims to several very important aspects. It's not only the characterization and the diagnosis of the respective leukemia and its subtypes. It also brings a lot of information on prognosis, for risk stratification and specific treatment. We today speak about precision medicine, about the respective therapy. We will also implement predictive information. And finally, and this is an increasing need, we are able to measure residual disease to control the therapy and its effects and to further stratify treatment accordingly. Now, leukemia diagnostics depends on several techniques. Some of them are very old, more than 100 years, like cytomorphology. Cytogenetics has been introduced in that field since nearly 50 years. We have histology that is normally done by pathologists, and I will not cover this subject because we don't do tree-fine biopsies, we don't do paraffin-embedded material. We work with liquids like blood and bone marrow aspirates. Then immunophenotyping, FISH is also able to get a lot of information on the respective genes that are destroyed, gained, or lost. And finally, and this will be the most part of my talk, molecular genetics, this increasing field in leukemia diagnostics and in many other subjects that we deal today. Now, the WHO classification was published in the last 20 years 3 times, in 2001, 2008, and 2017. And this is the Bible of the classification and diagnostication of leukemias and lymphomas. And what we see here is that over the last 20 years, we have an increased number of so-called genetically defined entities. There are some subjects of acute myeloid leukemia, myelodysplastic syndrome, myeloproliferative neoplasms, and acute lymphoblastic leukemia. And this number of specifically genetically, molecular genetically or cytogenetically defined entities, this number will increase over the next 5 years. In addition, so what we have to have in mind today is that during the last years, and this is just a compilation of some of the names, a lot of drugs were approved by the FDA, we can call targeted drugs. And there are much more in 2020, and there will more coming. And these drugs follow specific pathways. They address specific checkpoints, they address specific targets within the malignant diseases, and we can order them according to their mode of action. And we have a lot of kinase inhibitors like TKIs, we have monoclonal antibodies, and many other different approaches to treat leukemias and lymphomas. So just 2 examples, what happened in the last 30 or 40 years in leukemia diagnostics and treatment. One very good example, and you may be aware of this, is the chronic myeloid leukemia. We know that there's a very specific cytogenetic change, the translocation 922. There are specific genes that are involved, BCR and ABL1, and we have a translocation. And this translocation between BCR and ABL1 leads to a specific fusion gene, and this leads to specific changes, and the malignant cells start growing. And this is a slide from the German study group of CML that gives us an information about what happened in the treatment of CML, and this is all CML during the last years from 1983 to this year. And what we see is, due to the introduction of new treatment approaches, and TKIs for sure are the most important ones, the same disease now has a very stable over 80, over 90% of survival. And there are many studies already published that demonstrate that these patients have a normal lifespan when they are treated correct. And another subject I want to demonstrate here in the next slide is AML. So this is very from the Timothy Lay paper from the New England Journal 2013, and we see here a very clear-cut overview of cytogenetics and molecular genetic changes in AML and the respective prognostic information. But what we see here even more is that there are more and more targeted drugs available that targeting very specific subjects of AML changes, like FLT3 inhibitors or all-trans-retinoic acid and arsenic trioxide to cover PMRARA, fusion genes in AML patients. And this is very important because without looking into these changes on the genetic and molecular genetic background, we will not be able to apply the correct and the best drugs for patients' treatment. Now, the most important thing that brings us all these new information from research point of view, but also now in routine, is next generation sequencing. This technique is really a revolution in any kind of diagnostic approaches in leukemia and many other diseases as well. And it starts with the DNA of the patient, then there's a fragmentation, there's a barcode that leads to very specific barcoded DNA of the respective patients. That means you can combine up to 386 or even more patients in one sequencing run. Then you do an amplification of the respective patient's DNA, barcoded. You do capture for those genes you want to really sequence. You do an enrichment for those genes that had been captured. And finally, you go for sequencing. The turnaround time for this procedure is quite short in these days when a lot of things have been established. And the beauty of this approach is that you can combine genes of interest. You can combine panels for solid tumors, you can combine panels for myeloid leukemias, or you can combine genes and panels for lymphatic leukemias. And the number of panels is increasing, and the number of genes per panel is also increasing. It ranges between 20 and up to 500 or even more. And the turnaround time for this procedure to come up with the final material to go for the sequencing machine is below 1 day. After that, you have a lot of data. I will come back to sequencing later, but after sequencing, maybe on any kind of next generation sequencing machine, you come up with the raw data, and these raw data have to be mapped to the normal reference sequencing, and then you come up with more information. With that, you have to apply several tools, and this is still very open aspect. So for sure, there are some softwares available, but from my perspective, there's an increasing need to completely organize this way. The sequencing is the easier part from my perspective from NGS, but the work with the data and write it in a report, it's much more complicated, and there are not software tools available that are completely free of mistakes or do this automatically. So we need to be aware that there's an unmet need for going into the data. So we do the variant calling, then we do the all these different aspects here. And finally, at the end of the day, we combine all these algorithms in our pipeline, and this is an in-house established pipeline to annotate these respective variants. We formerly spoke about mutants or mutations. Now we speak about variants. And then this interpretation has to be done. We use several tools that are available to get information on the respective findings that came out of the sequencer. And we have a so-called MLI predictor that combines all the knowledge that we gained over the last more than 10 years of using NGS in our laboratory in many thousands of patients. And finally, before any bioinformatician or any molecular biologist is going to present the data to be written in a report, we get all this information compiled from the databases that are available in the web and our in-house database that is quite huge. And then we go forward to write the respective report. And to do this step, we already implement in our MLL predictor artificial intelligence tools that help us by decision trees and even deep learning algorithms to get more information out of the data that are available and to not start with this interpretation from every single variant at the beginning. Now, one additional aspect is not only how to handle the data, but how to run all these bioinformatic needed pipelines. And this is one little example how we could scale up this interpretation, because time is very important. The turnaround time has to be short. And we think that all this information should be given to the doctor and the patient as quick as possible. So we, several years ago, calculated these samples, sample by sample, after they came back from the gene panel sequencing machine, 63 genes per patient. And if we, let's say, combine 80 cases per run, the calculation time to get it from the raw data to the data that you can use for the reporting per sample was 15 minutes. But although we had a huge local infrastructure, 80 samples had to be calculated one after the other. That means the last sample, the last patient at that day was much later available to write the report. Now we changed this, and we went with our new tools that are much more informative and much more helpful to get information, and the new panels into the cloud. Within this, the cloud takes combining all this information with all the available information in the web per case, 40 minutes. But we can completely open the space in the cloud. That means for 80 sample, it's also 40 minutes because it's done in parallel. And if it would be 500 samples, it would be also 40 minutes for all of them. And that really makes a difference. So this is just one little example that cloud computing and the combination of huge data will really shorten the turnaround time. Now, the science of today and a lot of things we are doing also in our lab is science is the technology of tomorrow. And I want to demonstrate to you what we are going to share in the next upcoming ASH meeting. That will be a virtual meeting in December. And we combine there our routine tools of the methods we use in daily routine, and in a cooperation together with AWS using AI technologies, we approach three different aspects. We use the microscope images that were taken. It's around 600,000 cells. We used AWS tools with machine learning, and we came up with an accuracy of 92% to define the correct cell. And I can tell you that the reproducibility of a technician or a doctor is in that range or even much lower. So we are just working on this tool further, but this abstract we submitted to ASH will be presented there as a talk. The next method we used is immune phenotyping flow. The raw data were put into an AI tool together with AWS, and the accuracy to predict the correct diagnosis on the raw data of the flow machines without any compensation and any other pictures in between is 97%. And finally, this will be a post at the ASH. And finally, we used, together with Meta Systems, deep neural network learning and AI to go from the microscopic image of a metaphase to a complete karyogram. By doing that, we reduced the turnaround time from 7 days to 5 days and from 5 days to 3 days. And this also will be a talk at the upcoming ASH meeting. So we think that we can really improve our gold standards that come up with phenotypes by implementing AI and cloud computing, and that will lead to a very high reproducibility and will completely shorten the turnaround time from the sample to the result and the report. We are going to test this prospectively, and within the clinicaltrial.gov platform, there will be a prospective trial. We call this Better Leukemia Diagnostics through AI, short form Beluga. And we'll just start in the next weeks to go for 25,000 cases from morphology and in parallel 25,000 cases from flow to understand what the gold standard of a technician and a doctor on one side and the AI on the other side will be. And we will work on that to at least be equivalent to the gold standard. Maybe we can even outperform. We will know better in 1 year because we think we can enroll these 25,000 cases each within 1 year in our laboratory in a prospective study. But this is not all. This was phenotype, this was morphology. And we think that gene panels will not be the end of the story, especially when we speak about precision medicine and AI and what we can put together in all these new techniques. So we think that we will move in the next 5 to 10 years from gene panels to genomes, exomes, and transcriptomes. And to do this, we set up a pilot study in our laboratory, just in the research area, and we implemented a lot of our materials into genome sequencing. Why is this possible? Because the machines are scaled up in the last years, and if we use the biggest machine and use the biggest flow cell, the S4, we can run up to 250 exomes per run in one flow cell. If we cover a genome 30x, we can run 24. If we cover a genome 100x, what we did, we can run 9 per run and 200 transcriptomes. And what is fascinating for me is that the runtime is below 2 days. That means you can really scale up sequencing by using these new technologies, these new flow cells. And we are doing this now for today in a research setting, but I think it will change in the next years to routine. And this is the machines we use. We have 5 NovaSeqs in our laboratory to run all these research. And I can tell you that they are already now also in the routine. They are used for the panels, and that can be combined in one flow cell. So there's a lot of improvement in the technologies for routine and for research and or for research on the same machines. Now, the turnaround time for a genome, as we thought before, would be quite different from a panel sequencing or morphology, but the turnaround time of a genome also changed. So the workflow we started to implement in our research and intend to implement in our routine workflow in the next years is set up the way that we start with the DNA and the RNA, we go for the respective processing steps, then we go for the sequencing, and then we go for the data procession, and finally we end up with the turnaround time below 1 week. And I think that's quite fascinating that a genome could be sequenced in such a short time, including all the data processing and on the way to write a report. And what is also of interest is that we are completely accredited also for genome sequencing, including cloud computing and AI. So we follow the ISO 15189. This is the highest accreditation rules in Europe. And the complete workflow I show you here on this slide is accredited according to this ISO norm. What we did in the last 2.5 years is we sequenced overall more than 5,200 genomes with the respective diagnosis, as you can see down here, nearly 800 AML cases, more than 700 cases with MDS, and so on, taken from our research area and our freezers. And we have an overlap of 4,600 cases where we have the complete information of a genome and a transcriptome. And as we need a very high sensitivity in hematology, the median coverage for all the genomes is 103x. So it's much higher than what you normally expect when you speak about whole genome sequencing. We reach a sensitivity here with the genomes in the area of 1 to 2% mutations in the respective genome. Now, I would like to share with you 3 examples that we know already from treatment, targeted treatment, that we know from new drugs that are used in the field of precision medicine, and what we can see in our genomes. This is a little bit looking backwards, but if we could address our genomes for new pathways, this will be one of the ways everyone is looking to find new drugs and new targets. Now we know about BCL2 overexpression, and we know about this drug venetoclax that has been introduced in that field. And there are a lot of diseases that can now be treated with this drug, but one disease is CLL, especially those that harbor p53 mutations or deletions. And this is now the data set that we have in our genomes. And we just look here on the BCL2 expression in those cases with CLL, and you see this huge overexpression here in the CLL cases. And in addition, you also can see that other diseases like diffuse large B-cell or follicular lymphoma or mantle cell lymphoma, they also demonstrate an overexpression of BCL2 in the respective genome used in the transcriptome. So that demonstrates that things we know already can be reproduced in our transcriptomes from all these cases we investigated in the last 2.5 years. We have to have in mind, and this is also what we have to do, that there are already some mechanisms of resistance to BCL2 inhibitors. So we have to check this as well, but not on the genomic level, but on the respective gene level. Now, the second example refers to hairy cell leukemia. We know that hairy cell leukemia has a specific immunophenotype, but it also has a specific mutation. In nearly all of the cases, a BRAF mutation, a V600E. And the next slide here demonstrates to you the transcriptomic data on hairy cell leukemia here and splenic marginal zone lymphoma, or the hairy cell leukemia variant here. And you clearly can demonstrate here that we can completely separate the hairy cell leukemia with the normal BRAF mutations on one side and the other, quite closely related diseases with respect to their immunophenotype on the other side. And there's a specific hairy cell leukemia located here, but you see that the mutation we found here is different from the one that is detectable in all the other cases. Now that demonstrates that you can discriminate those diseases based on their transcriptome profile. The third example refers to multiple myeloma. And over the last 60 years, there was a lot of increase with respect to new drugs, and especially the last 10 years is really revolutionizing the treatment of multiple myeloma. And there is one very important pathway that addresses the respective plasma cells, the malignant myeloma cells, with specific monoclonal antibodies. And several drugs are already available and are approved by FDA and EMA. And I would like to demonstrate to you two findings that refer to CD38 expression and to SLAMF7 expression. And if we just know that these two targets are addressed by specific antibodies that are linked to these structures on the multiple myeloma cells, this is the data set taken from our genomes. And you can see here, all these are multiple myeloma cases with respect to gold standard diagnosis. And all these cases here are other lymphocytic entities. And what is discriminating here is the expression of CD38 and the expression of SLAMF7. And that again demonstrates to you that we can really pick up these findings that are now used already in treatment on the genomic level, on transcriptomes or whole genomes. Now, what we think is that the combination of the gold standards today, like cytomorphology, immunophenotyping, cytogenetics, FISH, and panel sequencing by molecular genetics may move over to transcriptomes and genomes, because we already can pick up all the structures that we see in genetics and molecular genetics with the so-called gold standards on the genomic level. And we think that this will be the diagnostic of the future, not only to pick up new targets and deliver new information on precision medicine, but also to make any kind of diagnostics more reliable and reproducible. So, and we think that these testings might not happen only at the time point when there is a tumor and you take a biopsy and you go for testing of a genome or an exome or transcriptome, but it may start at different time points of the lifespan with prenatal testing already. And there are a lot of reasons to think that that will move into the future, test all this. Now, there are some companies and some ideas already out, and you may all have seen that Illumina acquired Grail some days ago to go for detecting cancer very early with a simple blast test with liquid biopsy. And we in the hematology field normally do not refer to liquid biopsy so far. It's more the domain of solid tumors, but I would just like to share with you some unpublished data here. And these are cases with MDS, and we know about their molecular variants and their mutations in the respective genes. Here are the genes of the respective patients from left to right. We know about them when we investigated the mononuclear cells. So what we have here, the peripheral blood and the respective variant allele frequency in the genes, we have the liquid biopsy material, and we have the bone marrow. And what you can see here is that in parallel to the measurement we did in the peripheral blood or in the bone marrow, we can pick them up all in the liquid biopsy as well. So there is a field in hematology in the future. Now, where should this all go? I think we are on the way to do personalized signaling models for targeted treatment. We will start with some information from the literature and from databases. We have a generic model, and then we are going to really understand the effects of any kind of targeted therapies. We are going to reveal the adaptive behavior, and we are going to look for more information about any variables that may change and we cannot measure normally. And finally, we are going to model downstream processes and phenotypic responses. And I think that will lead, in combination with the patient-specific data that we can gain from the genome, from the transcriptome, even from the proteome, to very, very specific treatment approaches in the future. I have to admit that there's a challenge to make those models patient-specific based on RNA sequencing expression profiles only, but this is the way we feel it will go. That means we have the somatic cells that are benign, and we have the genetic change that leads to the malignant diagnosis. And then we go to any kind of dysregulation of cellular processes, pathways that are disturbed, and we see that there's the tumor. And by looking on the altered transcripts we see on the genome and we see on the transcriptome, on RNA level and the overall underexpression, we can go forward. And we see these transcriptional changes, and we can discriminate by the information taken from the genomes or the transcriptomes. We can discriminate between tumor and normal cell, and that will lead us to more information on the mechanisms and how to treat those patients better than we do right now. We did that with patients all run with genomes and transcriptomes in our study, the 5,000 genome study, and we checked between those patients who responded to treatment and who did not respond to treatment having an AML. And what we can pick up were 3 different makeups in the non-responders, because we found out somatic mutations and SNPs together located in areas where there are normally drug targets. And that leads to, for those patients who did not respond to the problem. We also picked up SNPs in combination, association of more than one, that with respect to drug target genes changed the response of the patient and led to non-response as far as we can predict right now. And finally, we had a third step where we see SNPs in the 3' region that modulate the mRNA interaction. And there are many things we can pick up from the genomes by using these techniques. So we think in the future, there will be a lot of things that we can do in a virtual approach. And we use the information of a normal cohort or of a cohort with malignant diseases. We use the genomics and the transcriptomic information, or even the proteomics. I think this is a little bit far behind, but it may be of interest for sure. We use all this omics data, And then we use in parallel databases from any kind of molecular interactions. And then we try to put a new drug and a new algorithm and a new approach to a specific pathway into this model. And we define specific population-based individuals that may be addressed by the disease and by the new drug. And we take virtual patients. We build up a digital twin and finally try to find out what would be the best drugs to address this change in the genome or the transcriptome. And there is already an option to build up in silico clinical trials, because we all know that phase 1, phase 2, phase 3, and so on, takes a long time to do. And we think that on this new approach, there might be a lot of help to save time and to get to new drugs and new pathway drugs that go for precision medicine, for getting it closer to the patient in a shorter time. Now, finally, and I come to the end of my talk, we see here that this is not all. This is not only the phenotype, this is not only the genome, this is not only the transcriptome, but, and I take this from a paper from Eric Topol, this is much more that we have to have in mind when we go to really understand what a patient's needs are. And all this knowledge is of interest to not only define the disease, but also define the patient and the patient's biological background. This is located in the genome and the transcriptome, and not only in the tumor cells, genome and transcriptomes. And I think we need it for sure, AI and a lot of cloud computing infrastructure to combine all this for the best diagnosis and the best treatment. And this is again the word precision medicine, to cure more patients or at least to prolong their lifespan. So, and I like to quote Eric Topol, and he wrote this great book from my perspective to introduce the deep medicine approach. And he wrote there, and I think that is very important for especially the younger doctors and younger scientists worldwide, AI will not replace physicians. However, physicians who will use AI will replace those who don't. And this is now the time to really move away from our standard procedures we did so far. We need to move away from phenotype to genotype. We need to move away from our laptop or our computer in one place to cloud computing and to combine this with artificial intelligence. Now, I would like to thank all my people that work with me in my laboratory, Claudia, responsible for cell genetics, and Fish, Wolfgang, for immunophenotyping, and Maya Megendorfer is the head of the molecular part and is also the head of research and development, and three of our bioinformaticians you see here, Wenke, Niroshan, and Stefan, who work in that field of daily routine implementation of any of these new tools and get them for accreditation, and also within the 5,000 genomes for research. And finally, I would like to thank the complete team of my laboratory. Without them, we couldn't do all that, combining routine and research to better serve doctors and patients' need and the time when we have all these new drugs, we have all these new options in the laboratory, and to bring this together by the use of AI and cloud computing. I would like to thank you for your attention, and I'll be very happy to take your questions.
Many thanks, Torsten. And please start asking questions. We have already one question online, and that is, I read it to you, wondering how a future user-friendly readout could look like, facilitating the linkage from the complex diagnostic findings to the drugs most likely to work could look like.
Yes, good evening also from my side. Before I answer the question, I can tell you that's hard to listen to your own talk that was recorded a week ago. But anyhow, I survived. And the question is very good because, as I told you before, the gold standards are not as easy as we all think. The reproducibility and the accuracy is far away from 100%. we are moving into the area with the molecular genetics and any kind of molecular discrimination and description of leukemias and lymphomas and all other diseases by the use of NGS and maybe by the use of genomes. But the bottleneck still is how to handle the data, how to get rid of all the noise, how to get rid of all these information that is not really meant to be important for the respective disease or for the respective treatment. And there are already some databases that try to not only give the best diagnosis, but also to give some information on the best treatment. I have to admit this is at the beginning. There are some databases that address this in lung cancer, and there are others that address this in other solid tumors. But with respect to leukemias, we are far away from 1 + 1 is 2 decision and solution. But we are working on that, and that needs to be also updated all the time, because there will be new drugs for new diseases and for new time points to use a drug at diagnosis or as relapse. And the final tool, and this is what we all would like to have, is get the information from the diagnosis in one tool without any contradictions, what is delivered to doctors and patients, and get the advice for the best drugs at that moment. But I have to admit, I think it will take some more years to get that done, but it will be possible, and we are on the good way.
Thank you. So while we would encourage the audience to type in more questions, Torsten, may I ask you one? Something that always worries me about artificial intelligence is that we don't understand it. It's usually run as a black box approach. And we have seen, some curious examples, but also some really bad examples where systematic racism has pervaded US healthcare delivery, et cetera. And the fact that you put a huge amount of data into a black box and then you get something structured out of it, but you don't really fully understand how it works, is what makes me uneasy about it. So what approaches do you and your teams use to address this, and how do you see that?
Well, this is also what I'm afraid of, because I'm a doctor and I want to treat patients, and I don't just play around with some new algorithms and produce a new video game or something like that. So we and others combine our knowledge from the gold standards, and this is baseline knowledge. I demonstrated at the beginning of my talk the WHO classification. We combine this knowledge and we combine all the data we have from our gold standards and look into the genomes back and forward. So we look into the orthogonal approach. That means, first of all, when we should really believe what's in that genomes coming out from artificial intelligence approaches, or even from easier approaches like I define right now, I tell you right now, is do we really pick up what we know before of this patients, of this leukemia in the genome and in the transcriptome? This is the first proof of principle, the first proof of concept that this will work. We are quite advanced in this, and we are quite sure that we can reproduce the needs and the classification of gold standards leukemia diagnostics from today in genomes and in transcriptomes. The next step is, and this was one of my examples from these, for example, expression of CD38 and SLAMF7, can we pick up information in these genomes and transcriptomes that are already used in precision medicine? And if this is not possible by AI tools, then we would also not follow up this approach further. And the third thing we are doing is we use a lot of different machine learning algorithms together with our team, working hard on that. Together with AWS, we also had, as some of you may know, a cooperation with IBM on that field. So we also work with other parties, other companies that are well known in that field of AI. But we only believe what we really can bring back to the final information we know already from these patients and their leukemias. I have to admit, Axel, that you have to be sometimes quite optimistic, but I am. And the reason that we don't know everything what's going on in an AI classifier should not lead to the final conclusion that we wouldn't follow this idea. Right now, we don't use it for any clinical decision-making taking from the 5,000 genomes, but we already use, together with our team, the information from cytogenetics and metaphase karyotyping together and combined with AI tools. This is pattern recognition. So we are just on the border of machine learning and personnel, and we will never get away from this when we are not quite sure, and we are at the beginning to transform this to more machine learning and less personnel.
Thank you. Ruth, will you take over for the next question, please?
Yes, sure. So, I mean, there's actually almost two questions that, like I was going to ask, are very pragmatic. So, one coming in about, deep learning and AI changes diagnostic procedure, and but that evolves further over time. However, for those of us who life is trying to deliver clinical trials, and particularly in a randomized trial, it's important to have a constant diagnostic procedure. So, how would you address that?
Yeah, that's a very good question. And we try to convince pharma companies to think about it because this question has to be solved. As we are in our laboratory also do a lot of standard reference laboratory work for international pharma companies and pharma trials and receive samples from all over the world. So far, we realize that the techniques we should deliver are our gold standards: morphology, cytogenetics, flow, molecular genetics, maybe one gene or some genes. And we can convince now all the pharma company people to at least freeze down some of the material in the freezers for any other question that may come up in the future. Because we know from FDA and other bodies like EMA that and I think this is correct, we should try to define patients best for the respective drug and not use a new drug for all the patients with the disease, but try to find out the molecular structures that work best for the respective patients. But if you don't measure this during the trial and during a trial that recruits for 3 or 4 years, you will not be able to answer this question in 4 years from now. So at least you need to freeze down material to look for molecular targets when you are really going to see who will profit best. What we would like to do and what we think also is feasible, use the gold standards to start with the treatment and use the genomes in parallel from the same patients to find out more. So if I would be the one who had to decide a new trial, I would think about a strategy, not only use the baseline necessary gold standards to start the trial, but also to implement already from the beginning information with genomes and transcriptomes and exomes. And we do a trial like that with the colleagues from Würzburg in multiple myeloma. We already enrolled prospectively 100 patients. And we know already from those colleagues that they gained a lot of information on the first 100 patients with respect to even better treatment that they normally would apply when they use this trial drug that is now tested. So we need to move over and implement this thinking and these methods because they are available and they can be delivered in a very short turnaround.
I mean, this turnaround is very impressive. And we've got a very practical question from John Smythe, who's chair of our CDDF board. Can you comment on the cost of these sort of techniques if we're going to implement them?
Well, I can just comment on normal prices and list prices. If you think about the machines, you can just look into the web and you will see these machines where you want to go for sequencing a genome or transcriptome or an exome and do this on a large scale. These machines cost $1 million each right now. If you now think about the consumables, the 30x genome will be around $1,000 or euro. It will be cheaper in the next years. We have to cover 100x, so it's more expensive. Having said that, I have to add that at that time point, you don't have no calculation on personnel that has to be skilled and very experienced, and you don't have calculation on the cloud computing and the runtime for getting the information out of the raw data. But I can tell you that looking into the complete cost model, without computing and storage in cloud and runtime for getting from the raw data to the VCF files is the cheapest price part from all the complete procedure.
Thank you. Axel, back to you.
Yeah, thank you. Another question here is, could you please elaborate on your results with liquid biopsy in MDS? What material was actually used, and does this require a specific AI approach?
We used the peripheral blood from patients, and we separated all the mononuclear cells that we normally use for our approaches, the hematological gold standards, and used for the liquid biopsy, the serum from the respective patients. We cannot exclude that there may be 1 or 2 granulocytes in that, or some of these leukemia cells, but we had a lot of controls and spent a lot of time to set this up. What we see right now is that we, as I demonstrated on my slide, we can pick up these variants, these mutations in the cells of the respective leukemia or MDS in this case. Also in the other compartment, the liquid biopsy. But from my perspective, it will not be the method of choice in hematology for the future, because we normally have these mononuclear cells where we know that the tumor is in. So I see a much broader field to use this in the solid tumors, and there many other people are already using it, like let's say in lung cancer. And the company Grail is doing the same. They work with peripheral blood to find out if there's a pancreatic cancer right now or in the future. So if you would ask me today, there are some very interesting papers for lymphomas that are even outperforming PET scans by using liquid biopsy in lymphoma patients. But for MDS, I think it will not be the method of choice. But there are a lot of things ongoing, and maybe in 3 to 5 years, I will tell you another story about it.
Okay, Ruth, do you want to?
Yes, we've got a question here. Could you comment on the feedback loop used in your approach, which allows you to, or would allow, to improve accuracy beyond 91%?
Yes, so with respect to the morphology, I think this is the question what is asked. Yeah, so there are, as you know, machine learning is always learning. So the tool we are using right now, and that will be also tested in the prospective trial, is that the machine learning model will name the cells of interest and will tell us from the machine learning perspective, this cell is this cell. It will be given an accuracy how sure the AI tool is that this cell is a cell. It will also tell us the second best choice and the respective accuracy that this cell is the other cell. So we can combine these tools by just saying, well, if you're sure with the accuracy above, let's say 93, 94, 95%, don't ask me, just go ahead. But for those who are below, show me the respective cells, and I try to name it again or tell you that you are correct already. So it's an interactive tool to improve the accuracy in the respective algorithms. We may also add some more cells for the improvement of the algorithms, but I'm not quite sure that will really make a difference, because this algorithm we'll use right now, and we already use, depends on 80,000 cells out of 600,000 we have. And even the very rare cell types, we have more than 2,000 or 3,000 each. And so at the end of the day, when you describe a phenotype, you will never be 100% accuracy. You will never reach that. And this may not make a difference. What really is important, if you need to pick up the malignant cells, and with respect to the blast cells, our classifier is 99% correct. And so you always have to check balance and have to check what you really need to achieve to serve patients best. And I think we are on a good way to even improve what we already achieved by the technique I just described you before.
Thank you, Torsten. I would like to combine the next question with a question that I have. So the question is, deep learning artificial intelligence changes diagnostic procedures as it evolves over time. And for me, and we have discussed it from time to time, I think this is a fascinating thing, because if you allow machine learning on such a massive set of raw data, it's likely, I think, that some of the classification will change. And we know how often the classification of hematological diseases has changed and how beneficial that was. So how do you see this? How do you see that this influence will change? What
Yes.
maybe be in 5 years or 10 years between the blue covers of the WHO classification?
Well, that's a good question, and I'll try to answer this. So we have to accept that a classification is based on humans. It's based on what you count. I give you the example, and Axel, you know this. We call a disease an MDS when the blast count is below 20%. You call it AML when the blast count is 20 or above. What does it mean? Nothing. It's really difficult to reproduce 19 or 21, but if it's 19, it's an MDS. If it's 21, it's an AML. And there are a lot of other classification regularies that we use, and we have to use it. That's okay. I don't have any better idea today, but a huge data set as we have right now, and we will increase the data set, may come up with better ideas, may come up with the idea that this looks the same and this looks the same, irrespective of how we call this disease. And this may be very important, because we would, at the end of the day, not be interested how we call this disease, but we would be interested how to treat it best. And if we could combine the information on drug availability and the respective infrastructure in the respective genome or transcriptome, I think everyone would be more happy than just name the disease as correct as we do when we apply all the rules from WHO classification. So we have to really rethink how the classification rules work if you go from phenotype and definition to genotype based on genomes and transcriptomes. And I would be very happy if we go more for pathway regulated classification than for any definition that was done on a table next to the microscope. That was correct the last years. It will not be correct the next years.
Thank you very much.
So, I mean, following on almost from that, this is bringing us very close to true personalized treatment, for example, in AML. Can you elaborate on how machine learning might change the design of clinical trials in new drug development for AML? There's a follow-on question of that. The final one on the chat is, CT, PET and MRI scans, will they become obsolete? So how is it going to change design, and will we not be scanning?
I will have so many enemies at the end of this discussion, because I always say it will be obsolete, this and this and that. That's not true. It will not be obsolete. I think we need to test this back to back together in one study. And then we learned a lot. 15 years ago, PET scan was not the routine. Now it is. So we will learn a lot, and we will see where's the respective place. I'm sure that some things will move away, because they, at the end of the day, will not be will not have any advantage in comparison to the new techniques. But coming back to your trial question, so we all know how cumbersome, how long, and how expensive it is to run phase 1, phase 2, and phase 3. And we know already that there's a lot of drug design is ongoing to find out what drug could be used best, and not do this in all animal models and all cell line models and so on. And what I told in my talk as well is that I'm quite sure that something like a digital twin from a patient or from just a man, a human being, will help us to understand where the infrastructure should be to be addressed by any kind of drug. And again, here, I'm sure that the standard routine to test a new drug in a huge number of patients. We just now see this in COVID, how difficult it is, even if all the world tries to do it best. So we have to do this trials still, but again, here we should get to the data on the genomic level and then try to put this in AI and machine learning and cloud computing. It wasn't possible 5 or 10 years ago, but now it is. And with this in parallel, we will learn how to get away from the structure we used to develop new drugs over the last 50 years to maybe more smarter approaches where we use all these options that are now available by using the tools I addressed in my.
Thorsten, at least my final question, following up on what you've just said, if I had a drug that would work in AML, and I've done a traditional phase 1 study, I know the dose, and now I would planning my phase 2 design and come to you and say, okay, so I don't know exactly, what are the subgroups. I know the molecular target, I know it works in AML, but so how can I use my phase 2 study to plan the best development program, to understand best from these new technologies how to run a phase 3 study, how to define the population in which the drug will be best? What would be your answer?
Yeah, well, we and others, we have now more and more data available. I have to admit, and this is true for everyone, that the data that we have on the genomic level or on the transcriptomic level are better when we really know what the patient's disease was before. So without a good description of the gold standard methods, we will not be very successful. But if we have very well-defined subgroups of patients and data, and then we add the information on the genomic and transcriptomic level, we could address these rich and huge data sets by the knowledge that the companies have with their new drug, and address the respective pathways, address the respective overexpression or underexpression of some targets and tools. And this could be all done in silico. And before you go forward, we could check this. This is also true if you just think about starting a phase 1 trial and you want to know how often this is in leukemias or in lymphomas. And if we go for this disease, is there a specific infrastructure in other leukemias as well to run several trials in parallel and not one after the others, what normally is done? So I think all these genomic data that now are available worldwide in scientific community are very helpful to shorten the process of getting to a better drug. And I think everyone will be happy to help and share.
Thank you. That brings me to the end of my questions, at least for tonight. Ruth, anything from you?
No, nothing further from me either. Thank you.
So, Thorsten, first, we would like to thank you very much. There was also a very kind comment on the Q&A to that at least one participant considered it time well spent, but I will join him in my words. So thank you very much. We would also like to thank everyone who stayed with us for this hour in the evening. You will be mailed a survey for feedback. This is, of course, very, very important for us. I think we got good feedback on the first webinar, and it helps us to learn, helps us to understand what kind of meetings we should plan for the future. So please let us know what you think. And please don't forget the workshop that Ruth will be sharing in September next year, hopefully then face-to-face, where we'll explore these topics in much more depth and a lot of interaction. So thank you, Thorsten, thank you, Ruth, and good evening, everyone. Goodbye.
Thank you.